Okay, I'm assuming you can see something that looks like some slides.
We see a slide.
Okay.
Yep.
Yeah. Okay, great. So yeah, as Suzanne said, I wanted to talk about hang on, I've just got a notification to say my screen sharing is paused. Can you still see the slides change?
No.
I've not seen the slide change, no.
Okay, hang on, let me find out what's happened here. Okay, let's try this again. Okay, I think that might be it now.
Yeah, you look live.
Yeah, okay, good. Yeah, sorry, for some reason it kept saying your screen sharing is paused. I've never seen that one before. Anyway, yes, I wanted to talk about the tools we have for exploring single cell RNA sequencing data in ViewPathDB. I'm going to do a demo of the tools that we have. And as Suzanne said, there isn't a walkthrough exercise in this session because we probably won't have time for it, but there is some homework. So there's an exercise available that you can look at in your own time if you want to. And that exercise will use the same data set that I'm going to demo today as well. So you should be somewhat familiar and it will cover some of the same ground. Okay, so before I do the demo though, I just wanted to talk very briefly about why we want to do single cell sequencing and a tiny little bit about how it works, just to set some context. So most biological samples are at least somewhat heterogeneous. Obviously the big picture case would be where you have a tissue, something like, I think this is supposed to be a brain, which has lots of different cell types in it. Some of the compartments in the brain can be very, very small and it's hard to dissect them out cleanly. And even within a compartment, like even within this blue compartment up here, you can imagine that the cells in the center are slightly different from the cells which are in direct contact with the green cells at the boundary here. Even if you're not working with a tissue, it's likely that you still have heterogeneity in your data set. Even if you're working with a single-celled organism or with cells in culture, it's likely that those cells aren't all doing exactly the same things at the same time. So it may be that your cells are going through cell cycle asynchronously, or they're differentiating asynchronously. Even if you synchronize one aspect of what those cells are doing, it's likely that they're asynchronous in some other aspect of their biology. And so even clean lab samples are not as homogeneous as you like to think they are. So when we do bulk RNA sequencing, the sort of RNA sequencing that we were looking at when you were doing the expression queries earlier today, what we're effectively doing is taking our tissue or our sample or our culture or whatever it is, and we're doing this with it. So we're taking all of those cell types, all of those different cells, which may be different types of cell, or they may be the same type of cell, but in slightly different states. And we're mixing them all together into a big pool, and then we're sequencing them. So when you do this, you can distinguish very strong signals. So I might be able to say here, if these colors were representing genes, that I might be able to say something about this blue gene. But weaker signals are masked, they're hidden by the stronger signals. And so, for example, in this image, I can only see one or two of these darker purple blocks. There's one there and there's one down here. So I probably wouldn't be able to tell very much about those from this sample. And the other problem is that we can't necessarily distinguish, if we do have a mixture of cells in our samples, we can't necessarily distinguish which of those cells are contributing which signals. We just get a sort of pooled signal. So it's difficult to associate signals with different types of cell in the sample. So what single cell sequencing allows us to do is to do something like this instead. So we can separate our cells out into different cell types, and then we can measure the expression of our genes in all of those cell types. And that allows us to do a couple of different things. So one of those different things is I can now say that the expression of gene A is only in the red cells, and expression of gene C is in this green population and this green population and nothing else. And I can see signals that I can attribute to these very small populations like this purple one here. That's something that those types of nuances get lost in bulk RNA sequencing. So by itself, just doing this is very useful in a lot of cases. But we can actually do a couple of things in some cases to take this even further and make it even more useful. So one of the things that is possible to do is what we call spatial transcriptomics. And that actually allows us to reconstruct how these groups of cells that we've signaled are physically related to each other, at least in 2 dimensions, as if it were, or actually is, as a section on a slide. So in the parasitology world, for example, you can imagine that that's incredibly useful for exploring host vector or parasite vector interactions. You can see which part of your tissue have parasites in and which parts don't, and look at what's happening with the host cells that are in contact with the parasites versus the host cells that are not. The other thing that you can do that has been very useful in the parasite world is something called trajectory analysis. And for this, what we do is we order the cells. So the image here, they've obviously been ordered by incremental changes in color along this trajectory. In what we're doing with the trajectory analysis with the cells is a very similar idea, but we're ordering them by incremental changes in gene expression. And that is very useful for examining processes that can't be synchronized, and also for finding intermediate cells that are between well-established life cycle stages. So you can imagine that if this were Plasmodium and we had a trajectory going from, I don't know, let's say merozoites coming into the liver through to the start of, so these are all the liver stages here in different places, and then the intraerythrocytic cycle starts down here. The merozoites are fairly well described, and the cells in the intraerythrocytic stages are fairly well described, but maybe we don't know all of the little things that happen along the way, and lining the cells up like this enables us to probe into that a little bit more. So the way, well, there are several ways of doing this, but the most popular way is the general protocol is known as droplet sequencing. And the specific implementation of that I'm going to talk about here is the 10x chromium, just because that's the most common one that we're seeing being used at the moment. So 10x chromium relies on a microfluidic system. That's depicted in the top left up here. And what we do with this is we have an aqueous buffer, and we feed gel beads into that buffer. The beads are coated in oligonucleotides. They have kind of hairy coat on them, and I'll talk a little bit more in a minute about exactly what that oligo looks like. But the important thing at the moment is that it has a poly-DT on the end, or some other capture oligo, but most commonly a poly-DT. Into that, we feed in our cells at quite a low concentration, so that they sort of go in one at a time rather than in clumps. And as the cells and the beads go through the microfluidic system, we then feed in oil. And because oil and water don't mix, what happens is that the aqueous buffer forms bubbles in the oil. And we adjust the flow rates of all of these different things such that we end up with hopefully one gel bead in each bubble. And it's a bit like doing cloning by limiting dilution. You're trying to get the concentration of the cells such that when you capture a cell with a bead, you only get one cell. And by doing that, you're actually going to also find that quite a few of your droplets don't have any cells in them at all. So your cells are obviously going to contain mRNA. Once the cells and the beads are in the droplet together, you can allow your cells to lyse, and because the oligos on the beads have a poly-DT on them, they're going to capture your polyadenylated mRNAs. You can then do your molecular biology, part of which happens inside the droplets. And once you have barcoded oligos on the end of your mRNAs, then you can actually dissolve your droplets and just pool everything and then sequence it. So these oligos are really the key to making everything work. What we've got here is this big circle over on the left depicts one of those gel beads. And then this thing hanging off it is one of the oligos that's on that gel bead. So all of the oligos have a primer that you can use to start your sequencing reaction when you come to sequencing. They all have your capture oligo, which, as I said, is usually a poly-DT. And then they have these two barcodes in the middle, and these are the really important bit. So the first of these, which I've colored the same color as the bead, is a cell barcode. And this one, every oligo that's attached to this bead is going to have exactly the same cell barcode on it. So if we've successfully got to a situation where we have one bead associated with one cell, then every transcript coming from that cell is going to have this same cell barcode on it. So after we pool everything, we can still figure out which transcripts came from a cell together. The other part of this is the unique molecular identifier, or the UMI. And for this one, every oligo attached to this bead will actually have a different UMI. And what this allows us to do is to keep track of how many individual mRNAs were captured from this cell. So when you do a bulk RNA sequencing, what you normally do is do some kind of amplification step before you do the sequencing, and then you're inferring how much mRNA you had at the start by how much cDNA you have now. In this case, because every mRNA will have a different UMI associated with it, we can actually measure directly how many different RNA molecules we had at the start. So it's much more sensitive in that way, and you don't have the same amplification biases that you have in bulk RNA sequencing. So I don't want to go too much into the analysis about this. I'll talk about some aspects of the analysis as we go through the demo, but actually the analysis would really be a whole workshop in its own right. What I do want to very quickly do is talk about the data set as well that I'm going to talk about, again, just because it's easier to understand the concept. So the data set I'm going to demo is using this trajectory inference strategy to explore the transition between slender stage and stumpy stage life cycle stages in Trypanosoma brucei. So just for those who are not familiar with this biology, Trypanosoma brucei is the parasite that causes African sleeping sickness. And in the human host, it has two main forms. So one of those is this form that's known as the slender stage. It's so-called because it's long and thin. And that's the predominant form in the human host. That's the host that's the form that lives in your bloodstream. It replicates, so it's going to increase the load of the parasite in your blood. It's primed for metabolizing sugars that are present in your blood. but it can't be transmitted. The other form that you find is this stumpy form, and you can see that morphologically it's shorter, and that form is, it can't replicate anymore. It's an evolutionary dead end if it doesn't get transmitted to the insect vector. It's pre-adapted for metabolizing stuff in the insect vector, but it can be transmitted. And so what happens is, it was known for a long time that differentiation from slender to stumpy was based on a quorum sensing type arrangement. It was known that you need to have a certain density of the slender forms before you start getting differentiation to stumpy forms. And it was known that certain factors in the media could force differentiation, but it wasn't known which factors they were. You'd put I think it was a brain-heart infusion broth into the media and they would differentiate. But obviously that's got a lot of stuff in it and no one really knew what it was. So about 4 years ago now, the mechanism was discovered here. So what happens is the slender form releases peptidases. It's extracellular in the blood, so those peptidases can chew up all of the proteins that are floating around in the serum and make oligopeptides. Those oligopeptides are then sensed by this GPR89 ligand. And so the more slender forms you have, the more peptidases, the more oligopeptides, the stronger the signal. And once that signal crosses the threshold, then you get differentiation to stumpy forms. So that was great, but what happens after this ligand binding event was still not understood. And that's what this group were trying to understand in the experiment that I'm about to show you. Okay, so let me close that and move over to TriTripDB. So this data set is in TriTripDB because it's a Trypanosoma brucei data set. We use a third-party application to allow you to visualize and interact with single-cell data sets, a thing called Cell by Gene. And it's similar to JBrowse in that the code is maintained by someone else, but we can integrate it into our website. So the first question is, how do you find Cell by Gene? And there's a couple of different ways to do this. So the first thing most people are going to try is looking for a search. So if I go to the searches here and I search for single cell, you can see we do indeed have a search here for genes by single cell evidence. And if I click on that, I can choose the data set I'm interested in, and I can ask to get the results. And this is not actually doing any kind of analysis at the moment. This is here purely to give you an entry point to the Cell by Gene app. So this just lists this 8,738 genes are the ones that were represented in this particular experiment. And for each one, we have a link to explore in Cell by Gene. The other place that can be useful to get it is actually to go to our datasets page. I don't think we've looked at this yet. There's one of these pages, on all of our websites. If you go to the data part of the menu and then to datasets in TriTripDB in this case, we now get a list of all of the datasets that are loaded into this particular database. And as always, there's a lot of stuff here and I can search it to make things easier. So if I start searching for single cell, I can see my data set here. If I click on that, I get to a page for this data set, and all of our data sets, regardless of their type, have this page, and we can see the publication, we can see the authors. Depending on the data type, there may be other information. And down at the bottom, we then also have a link to explore this data set in Cell by Gene. So I'm going to go ahead and click on that. Cell by gene can be a bit intensive. So if no one's used it for the last half hour, it's going to have to start itself up. So it may take a couple of seconds to start up the same way as it just did here. So what we're seeing now is the cell by gene app, the cell by gene view, I guess, for the data set where the authors were exploring this slender to stumpy transition. So in the middle, we have what's called a UMAP. And the UMAP is basically a scatter plot where every point represents an individual cell. Unlike a normal scatter plot, the X and the Y axes don't actually relate to anything you can measure here. If you think about what's going on, for each of these cells, we've captured a large number of transcripts, potentially up to about 9,000 transcripts in this organism. If we were to try and plot that all on one plot, it would be very difficult for a human to interpret. We're not very good at thinking in more than 2 or 3 dimensions. So what the UMAP has done is it's tried to compress all of those thousands of dimensions into 2 dimensions in a way that we can understand it. And it's doing that by trying to put cells that are similar to each other close together. So while the X and Y axes don't necessarily mean anything directly, what we can say is that these cells over here that are close to each other are more similar to each other than they are to these cells over here that are quite different. On the left, we have some metadata about these cells. So some of these things are quantitative data, and the ones you see over here are counts of the number of individual RNA molecules that were captured in each cell and the number of unique genes that were captured in each cell. Percentage of those genes that were mitochondrial and the percentage of those genes that were ribosomal RNAs. Those are common QA metrics. We also have some categorical metadata at the top. And this falls into 2 categories. The first thing we have here is the replicate. And this is experimental metadata, I guess. So if I expand this, I can see that this experiment actually had 2 replicates for wild types. And some of those cells, obviously one cell can't have come from 2 different experiments. So some of those cells will be from wild type 1 and some will be from wild type 2. And for anything like this, I can click on this droplet icon. And that's now going to color each of my cells. So now the replicate 1 cells are in purple and the replicate 2 cells are in green. If I mouse over either of these, you'll see that what it does is it redraws the purple points slightly bigger and on top, so they sort of pop out at you and you can see them. So if I flick between these 2 things, you can see that actually the overlap between these 2 replicates is pretty good. Maybe the green one has slightly fewer cells over in this cluster on the left-hand side, but they're not terrible. They're quite similar to each other. The other thing we have is experimentally derived metadata. And in this experiment, that's this cluster over here. The metadata that's present here will depend on what the authors did when they analyzed this data set. So in this case, the authors have done some clustering on this data. And they have derived 4 clusters. And again, I can click on the droplet icon and I can now this UMAP is colored by the different clusters. And just to make it easier for me to see what's what, I can also add the names using this button up here. So in this particular experiment, the authors were able to look at the genes that were highly expressed in each of these clusters and decide what the clusters could be. And part of the exercise that's available for you is looking at that. So they had 2 clusters that they decided represented slender forms over here, the purple and the pink, and 2 clusters over here that they decided represented stumpy forms, the 2 green yellow ones. Just to go back, while I've got both of these open, if I go back and colour by the replicate again, you can see that if I have the cluster part of this open, that it's put these little bars in. And these are showing me the proportion of cells in each group. So the top one is the proportion of cells in the long slender group that are in replicate 1, and the proportion that are in replicate 2. So I can confirm here that these two replicates were pretty similar between in the slender form, and that there were slightly fewer stumpy form ones in replicate 2 than replicate 1. But again, overall, I'm quite happy with these two replicates. And you get the converse here now. So now the wild type here are coloured using the colours up here to again show you the proportions of cells in each of those two replicates. Okay, so we're looking at our cells, and we've been able to put them into groups. And that's fine, but that doesn't really tell us anything new at this stage. So the slender forms are fairly easy to grow in culture. Those are already well described and well understood. The stumpy forms are also well described and well understood. And what the authors were actually interested in here wasn't the slenders or the stumpies, it's what's happening in this part here as they're transitioning from one to the other. And these are the cells that are very difficult to capture in a bulk RNA sequencing experiment. So this was the whole purpose of doing this experiment to start with. Turn those labels off because it makes everything bright. So yeah, these ones here in the middle are the ones that this group were interested in. So they did a trajectory analysis, and that's that thing where they've lined up all the cells based on incremental changes in gene expression. And then they've done a differential expression along that line. So we can't do that full analysis in cell by gene at the moment. I do actually know that the cell by gene team are working on this. So maybe at some point we will be able to, but not quite yet. But what we can do is we can go down here and we can change the visualization. So this fate visualization is the visualization where they've actually lined up this data set. So now if I put the labels back on, we can see we now have the slenders over here on the right, going across to the stumpies on the left. And they were, again, looking at the genes that were, where the peak of their expression was sort of in this window along this trajectory. So the group found 6 genes, and they thought that they found one of them that they thought was particularly interesting. And I've already got the gene page up for this. I looked at this in the paper earlier. And this is a gene, let me go to the top of the page. This is a zinc finger protein family member gene. And we can actually see some things on this gene page that that help us understand why the authors thought this was useful. And the most useful things for this are actually in the annotation, curation, and identifiers part of this page, and in the user comments. So any user who's logged in can always go and add a comment on a gene and tell us a bit more about what they think that gene does, or where it's expressed, or link to a paper where the gene's been described. So these are the user comments for this gene. And there's 2 here that help us understand why this was the one they chose to explore further. So for context here, Trypanosoma brucei does polycistronic transcription, and it doesn't have very good control of gene expression at the transcriptional level. It actually transcribes all of the genes at the same level, and almost all of its regulation is post-transcriptional. So the first comment here was that this gene and a paralog are known to stabilize mRNAs. And obviously stabilizing an mRNA is a form of post-transcriptional regulation. The other thing that was interesting here is that it's also been described as binding to and regulating RNA binding protein 10. And RNA binding protein 10 has already been described at this point as being involved in this slender to stumpy transition. So just from this, it seemed like this zinc finger protein might be an intermediate factor between the quorum sensing mechanism and the actual regulation. So on the gene page, if we go down to the transcriptomics section, you'll see that we also have single cell RNA sequencing here. And if I open the top row on this table, this is the experiment we were just looking at. And you can see you get a nice mini view of our UMAP here. And now it's colored by expression of this gene. And I can interact with the histogram over here, so I can ask it to show me just where the most highly expressed genes are. And you can see that there is a cluster of those highly expressed genes over here. If you come directly to a gene page, you'll also find down here, a link to this gene in Cell by Gene, and also a link to get to the initial view of Cell by Gene that we were looking at. So this gene, we can probably look at this one a little bit more in the app. I'm actually, rather than clicking on this link, I'm going to do it a different way. I'm just going to copy the gene ID so that I can show you how to get to this. Let me put this back to the UMAP. Over on the right now, we have a gene search. So I can enter my gene ID in there and select it. It shows me it's found this gene, and I can expand it. The histogram is showing the expression of this gene in all of the cells in this experiment. And I can also, again, use this droplet to paint my UMAP with this gene. And I can interact with this histogram in the same way. So this is showing us the same thing as we were just looking at on the gene page, but because it's bigger, it's a bit clearer. We've got a peak of expression sort of here. There's some cells expressing this gene over here as well. And maybe that's fine. We don't really know how early they commit to differentiation. So that's doesn't necessarily mean it's not interesting. And if we go back to the fate map, you can also see that here as well. We do have some cells expressing this gene early on, but there's also a lot of the cells in this central part of this trajectory are expressing that gene as well. So the next thing this group did was to knock that gene out. And after they'd knocked it out, they did single cell sequencing again of the knockout. We also have that data available in TriTripDB, and that is this other row in this table below this. So if I open that, what we've got in this one is we've actually got the same two wild type replicates as we had up here, but they've been combined with the knockout as well. And the immediate thing you can see, if I just sort of go up and down between these, is that the shape of the UMAP has changed. So let's open up the UMAP for this, the integrated data set with the knockout, and have a look at that one in a bit more detail. So if I just go back and forth between, I need to put this one back into UMAP mode, between these, let's click that off as well and put it in black so that they're comparable. You can see that here we've got our slenders over here, and they're all sort of differentiating into stumpies over here. This one is laid out similar. We do have slender forms over here, and we have stumpy forms over here. The connection between them isn't as clear, and we've got this other weird sort of bobble of cells up here. So let's have a look. You notice now we've got more metadata up here. We've got the replicates, so 2 wild type replicates plus the knockout, but it's easier for us at this point to combine the 2 wild type replicates, and so we can do that using the line. We've got the wild type line or the knockout line. If I paint my cells based on those, we can now see quite clearly what's happening here. So the wild type cells are pretty much doing what they were before. On the right hand, we've got slender cells, and then they're moving around into stumpy cells. And we've got a few that are going up into that weird population, but not too many.
5-minute warning.
By comparison, the knockout, we've got you can see that they're sort of turning right in this EMAP. So at the beginning, they overlap with the wild type pretty well, but almost none of them become stumpies. And instead, we make this other weird population up at the top up here. If I open the cluster, you can confirm that this is happening. So we can see that actually none of these knockout cells are in the Stumpy A or Stumpy B cluster. Almost all of the members of this long slender B2 cluster are knockouts, and they're fairly even in the long slender A clusters. So what the group went ahead and did next was a differential expression, and we can actually do this in this app. So they wanted to ask what's in this group of cells up here, that group up here, what's different about those compared to the cells that are behaving normally. And we can do that, we can replicate that right in this app. So if I can use these checkboxes over here to select or deselect clusters. At the moment, everything is selected. So if I deselect everything and then select only the B2 group, and I have 1,046 cells in this group, and I can add them up here to a group by clicking where it says 1. And you'll notice it goes from 0 cells to 1,046 cells. I've added those into group 1. I'm going to add everything else, so everything except the B2, into group 2 the same way. So I'm going to select them over here, click on group 2 to add them, and now I've got 9,848 cells in group 2. And now that I have 2 groups of cells which have been populated, this button here has become blue and I can use it. And this runs a differential expression between those 2 groups of cells and shows me the top 10 gene or something, I think. Top 10 differentially expressed genes. So that doesn't take very long. You see we've got 2 things popping up here. So population 1 refers to the group of cells that we put in the box labeled 1, and population 2 refers to the group of cells that we put in the box labeled 2 up here. A nice test, I'm just going to colour these cells back in so they show up. A nice test here is to look in the ones that are upregulated in group 2. So group 2 is everything except this green group. And if I open that, it's showing me now the top 10, top 20 or so, maybe even more than that, differentially expressed genes. And one of them, I can see it here because I looked up these gene IDs earlier. This one here is the knockout. And that's good, right? We would expect that our knockout would be expressed at a much lower level in the knockout than it would be in the wild type. What's nice about this is when you color these by the expression of a gene, over on the left here, you can see these little histograms, and that shows you how much the expression profile has shifted. So you can see here that the profile has shifted to the right in the wild type compared to the knockout. That can sometimes be easier to see than looking at the colors in the UMAP. But what they were actually interested in was what was upregulated in this group, and that's going to be in the population 1 high set, because this is population 1. So if I expand that, close that one down again. If we click on some of these, so we can open them up and see the histogram and click on them, we can actually see that some of these, this one's probably a better example, they are expressed in this group, but they're also expressed in the slender cells entirely. This is happening because what it's doing is taking the average of all of these cells compared to the average of all of the other cells. And of course, the average of all of the other cells is going to be decreased because this gene is not expressed in the stumpy forms particularly. So it's not specific to this group, but it's still coming out as differentially expressed. This one here that I clicked on first, I actually happen to know that this one is a known marker for slender cells. So that one's almost exactly what's happening here.
Catherine,
if we go down a bit,
we're about at 1 o'clock.
Yep, I just had like 2 more minutes, if that's okay.
Sure.
If we go down just a little bit further, I can begin to find some of the genes here that were actually thought to be interesting by this group in their paper. So if I expand this one and paint it and just get the very highest expressors here, you can see there's some elsewhere, but there's definitely a cluster of these here. This is a topoisomerase, so it's involved in DNA winding or unwinding or replication. And it's interesting because we know that one of the phenotypes is that stumpies don't replicate, but slenders do. And it seems that whatever's happening with this weird population, they're definitely still replicating. And they may even be replicating aberrantly. It seems like they have this thing switched on more maybe than the other cells. The other one that they noted might be interesting is this is a non-coding RNA here, which again is, sorry, let me click that one off, mostly expressed up in this cluster, and they think that that one may have some further regulatory properties. So that was about all I wanted to cover. I hope I've shown you how you can get to this app and some of the ways that you can interact with it and explore different things in it, and replicated some of the results from the paper. As I said, and Suzanne also said, the homework for this, if you want to do that exercise, is exploring the same data set in a little bit more detail as well. So yeah, that was it. I'm happy to answer any questions.
There was a question in the chat about tumor biology and you know, whether this is used there, and I answered that yes, definitely. Here's a new.
Yeah, it's used in all sorts of places. Cancer biology is definitely a big thing there. Also things like rheumatoid arthritis. There's a big rheumatoid arthritis group here who are using it and getting some really exciting findings from it. Quite a few parasitology groups now are also doing this kind of experiment that this group have done here, and some are beginning to start using it to explore host pathogen interactions as well. And we do have one data set that's doing that in PlasmoDB if you want to go and explore that one. So yeah, it's really exciting technology. It definitely has its limitations, but it's unlocking a lot of new things that just get lost in bulk RNA sequencing.
Sounds good.